It is very important for the immune system to recognize antigens and destroy them. As you know, we have two types of lymphocytes, B and T lymphocytes. So how does each one of them recognize the antigen? First of all, we will start with B cells. B lymphocytes have on their membrane molecules called or receptors called immunoglobulins, Ig, or you know them by the name of antibodies. Each B cell has a unique and can produce only one type of antibodies in its life. However, it's very important for us to have millions of types of antibodies to attack millions of types of antigens. And that's why we have millions of types of B lymphocytes. An antibody is a Y-shaped protein, it has the shape of a Y, that is made up of 4 polypeptide chains. 2 heavy chains, the ones in the middle, which are long peptide chains, and 2 light chains on the exterior. This antibody is characterized by the presence of a constant sequence. This is a constant sequence or the constant region, and a small variable region through which they bind to antigens. So this is the antigen binding site. Each antibody has 2 antigen binding sites, and this here is the region of fixation of the antibody to the B cell. Once a B cell is activated by a certain antigen, it will proliferate and multiply, part of which will become plasma cells, and we said that plasma cells have a small nucleus and are rich in endoplasmic reticulum and Golgi apparatus that are important for protein production, and this is a protein. There are 5 different classes of antibodies: IgM, IgD, IgG, IgA, and IgD. So, IgM is a pentamer. Pentamer means that it is a one molecule containing 5 antibodies, so it has 10 antigen binding sites. IgA, on the other hand, is a dimer. It has 2 antibodies linked to each other with 4 antigen binding sites. Now, each one of these antibodies has a certain characteristic. We obtained that the highest amount or the highest percentage of antibodies in the serum is for IgG, while IgA and IgM come second and third, and we have a very small amount of IgE and IgD antibodies. For example, also, only IgG can cross the placenta. IgA is found in the secretions of the body, for example, in colostrum, which is the first milk produced after delivery, saliva, and tears. IgM is important in the primary immune response, which is the first time we attack an antigen, while IgG is found more in the secondary immune response, where it is the second time or the third time we fight the same antigen. IgE is important in allergy, while IgD is found on the surface of the B cells. Here also in your book you can obtain the different types or the different classes of antibodies. There's also something called a cross reaction, which is when the same antibody binds to the same epitope which is found on 2 different antigens. For example, this is antigen 1, which has a circular epitope that we label as Z. This is antigen 2, which has the same epitope, which is a circle, also labeled as Z. So the same antibody in this case can bind to 2 different antigens. This is called a cross reaction. The cross reaction is important in the principle of vaccination when we prepare a vaccine. So now we learned what is the way by which B cells recognize an antigen. What is the mode of recognition in T cells, in T lymphocytes? T cells do not have antibodies. They have another type of receptor called a T cell receptor. This is a T cell receptor. It is made up of 2 polypeptide chains only. It is fixed on the surface of the T cell membrane. It's also characterized by a constant region and a variable region. However, a T cell receptor has one antigen binding site, which is here in the middle, and has two HLA binding sites, and this brings us to the discussion of how are T cells activated and how do they recognize an antigen. Antibodies can bind to free antigens. When an antigen is free in the serum, having this shape which can fit here, it directly binds to this antigen. However, the T cell cannot bind to a free antigen which has the complementary structure. For a T cell to be activated, it should bind to an antigen which is presented to it by HLA proteins. So the HLA proteins present this antigen to the T cell in order to bind to it. Thus, T cells can never bind to this antigen alone unless it is presented to it by HLA proteins. The binding of the TCR to HLA antigen complex is referred to as double recognition, where for a T cell to be activated, it should double recognize the HLA molecule and the non-self peptide. So by this we know now that a B cell produces an antibody or an immunoglobulin which has a Y-shaped protein made up of 4 polypeptide chains, 2 heavy and 2 light, while a T cell receptor is always fixed on the surface of a T cell and it has an HLA or 2 HLA binding sites and 1 antigen binding site, and it can only fix to antigens presented to it by the HLA proteins. However, antibodies can bind to free antigens. Also, the B cell, once activated, it proliferates and releases this antibody into the serum. However, the TCR is always fixed on the surface of the T cell receptor.